Hello, everyone, and I hope that you're doing great. Now, today I'm going to run through biology as a crash course series. I know the examination is on our tails, and some of you are anxious at this time, but there's no need to be anxious. It's just that you need to prepare yourself as best as you possibly can and be comfortable. All right, so what I'm going to do is to give you some explanations and tips as I go along. Also, I'm going to add some information in the description in terms of links so you can watch some extra lessons that will assist you in different aspects as I go along. All right, I think I've covered about 95% of the entire syllabus, which is good enough for you to at least aim for that grade 1 or A. All right, so the first thing first is look at the division of the syllabus or the course itself, and the course is divided into 3 segments. You have living organisms in the environment, and then you have life processes and diseases, and then you have continuity and variation. Now, before I go into the specific topics, what I'm going to look at is take a look at graphs. All right, because I know some of you at this point are still not so good in terms of drawing a proper graph, what to do when you see a graph, how to interpret graphs, and so on. So the first thing I have to know what a graph is, and a graph shows the relationship between variables, and the 2 variables that we have to wrestle with are independent variable and dependent variable. So first one, independent variable, those must be placed on the x-axis. Again, independent variable, as the word suggests, it stands on its own. Nothing will affect it, but it can affect other things. So again, this is a variable that can be manipulated. That means when you are doing an experiment, you actually can manually change this type of variable. All right? So in other words now, you as the experimenter can adjust the independent variable according to producing a certain result or a certain change or a certain response. All right? That is you doing this. So again, what is important about independent variable is that independent variable causes another variable to change. Example of this is time. What change will occur over a certain time? What temperature is another independent variable. Temperature can cause something else to change, like example, evaporation and so on. So the more heat you have, you can have higher evaporation rate. And the other one is dependent variable. As the word suggests, it depends on other variable. And so again, the dependent variable must be placed on the y-axis. Again, this type of variable changes based on the changes in the independent variable. An example of that is the rate of photosynthesis. All right, I'm going to give you an example that will bring this out a little bit. Also, growth or growth rate, water loss, for example, in transpiration. What are some external factors that affect the rate of water loss? All right, I notice that these things are the things that are being affected by the change in another variable, which we call the independent variable. All right, so I'm going to bring this out in terms of an example. And so what I have here is 2 variables. One is saying volume of carbon dioxide in centimeters cube, and one is saying mass of glucose produced in grams. And I know, as you see this, you should know what topic this is related to, and this is photosynthesis, because the amount of carbon dioxide a plant receives will determine the amount of glucose that is produced. All right, these numbers, I make up these numbers, so these are arbitrary numbers. All right, and so what I want to note is a pattern right here. As the carbon dioxide increases, you also have an increase in glucose. However, at a certain point, the amount of glucose produced will remain the same. And there's a reason for that. And of course, we can discuss that later. All right, now the idea of plotting this information on a graph is that one, you must remember, when you're doing a graph, you must have a title. So a title is important. For example, this is saying a graph showing the mass of glucose produced for the volume of carbon dioxide absorbed. All right, so a title is important. You must label your axis, your y-axis and the x-axis. You must label them, must. All right, and you must use a suitable scale. I notice I'm going through all of this for your benefit in terms of what you should look out for in terms of marks. That's why a graph will be like 4 or 5 marks, up to 6 marks sometimes. So your title is important. Your labelings are important. Your scale is important. And why scale is important? Because the graph should be at least half of a page, going to 3/4 of a page, the least. A small graph, you're going to lose a mark for it. So when you use large scales, the graph will be bigger and fill up the graph page. All right, so for example, we have the variables. Again, mass of glucose produced, notice is on the y-axis, and volume of carbon dioxide is on the x-axis. So if you look at the values and you realize the graph start to shape like this, and then after a point, you notice the amount of glucose produced remains the same, and simple because no matter how much carbon dioxide they put in, some other factor will be limited. For example, light or water. And so therefore, the plant can only produce glucose at a certain rate and no more based on the other limited factors. All right? And so also you need to learn how to interpret the graphs. What is also important, the line must be smooth and the points that you plot must be fine and on point. All right, so notice there are a number of marks you can earn from just drawing a graph. All right, so that being said, now let's go into our specific sections, and we're going to go to section A first, talking about classification. And remember that in classification is the way of organizing or grouping organisms based on specific features. And the largest group is kingdom, ranging to species. You must know them. All right, so you must know specific phylum, example, mammals. All right, you must know common versus scientific names. You also need to know scientific names made up of genus and species as well. All right, so those are things you need to look out for. And just to give you a little insight on this topic, you must know that we start from kingdom, phylum, class, order, family, genus, and species, in that order, coming from very large to very specific group. A way to remember this is that King Philip came over for grandma's soup. Back in the days when I used to go to high school, I used to remember that King Philip came over from Great Spain. But of course, as we go on, we change things and also adopt new concept. But once you remember it, it will be good. All right. Now, again, a point to note is that it must know the specific kingdom. For example, the plantae kingdom, the animalia, fungi, including mushroom, bacteria. All right, and bacteria is a prokaryote, just to make mention of that. All right, you also need to know the protista kingdom, which include amoeba. All right, the specific phylum that you need to know is vertebrate, and vertebrates, they're also called chordata. Those are the ones with backbones. And you have the invertebrates, those are the ones without backbones. So please, you need to know those. You need to also know examples from different classes, such as class fish, reptile, insects, bird, and mammals. And what you need to know is also specific characteristics, such, for example, birds and mammals are the only warm-blooded. All right, the others will be cold-blooded. You need to know things like that. You need to know to distinguish between them in terms of those who have scales. For example, birds have scales on their feet. That's point you need to know. Some people miss that. All right. Insects with the different segments of their bodies as well. All right, and also the amount of legs and segmented or jointed legs and so on. So you need to know the characteristics of these classes. Please, you need to go through them. All right, very important. All right, now let's talk about some specific things that you need to pay attention to. All right, again, we talk about characteristics, and CSEC will test you on identifying visible characteristics such as hairiness, color, all right, shape, all right, venation, number of legs, and also number of wings, and also body segments, which I mentioned earlier. All right, and so you need to know the differences and similarities between organisms, such as insect and arachnids. You need to know the difference between those. All right, the amount of segments, and I see this was also a past paper for multiple choice as well. When they compare the different organisms. And of course, the insect will have 3 body segments versus the arachnid will have only 2, the number of legs and so on. All right. Antenna, no antenna, and all of those things. So please, number of wings, wings or no wings. Yeah. Very important for you to know them. All right. And so again, just give you a little brief information, different sections. All right. The next topic I want to look at is ecology and ecological studies. And very importantly, you need to know things like niche. A niche is a specific role of organisms in an ecosystem or environment, the specific role that organism is playing in that environment or ecosystem. That's what they call a niche. Habitat is where the organism live, and then a population, very important for population, it is the total number of a specific species. So we don't mix species to get a population. There's only one type of species. You add up together, get a population within that habitat or in that ecosystem. Community now is the interlink of different populations or group of populations interacting. And then ecosystem now deals with the abiotic and biotic factors. So it's a grouping of all the living and non-living aspect of the environment. That's what an ecosystem is and how they interact with each other. All right. We also need to talk about adaptation of organisms to specific ecosystems, such as those that live in water, which are marine organisms, those that are terrestrial, those that live on land, those that live on the rocks, for example, those that live in trees. They talk about like camouflage. They talk about adapting type of leaves. For example, cacti that will be in the desert, they do not have true leaves or large leaves. And so they have they lose the leaves to adapt to conserve water and things like that. All right. Some plants will have thick waxy cuticle to prevent water loss. Some of them will have small leaves or leaflets. All right. Some of them will have also prickles for protection as well. All right. So definitely you need to know adaptations of organisms for them in a specific environment. All right. Camouflaging is also another one for example, fish will camouflage and other organisms that live different places, whether it be land or in water. All right. And also, you need to know methods that are used to study organisms within the ecosystem. And such one here is called potters. And just give it an example of that because that one's a common one that is normally used. So what the potters is really a device that will have two tubes. One will allow the organism to enter. And of course, you can also suck on the other end if you want to do that. You don't have to, but that's a typical way to do it. So, for example, it's a small organism. Instead of touching them, you will suck on one end of the straw, and the small organism will be stuck into the jar. And notice there'll be like a mesh type of thing right here to prevent the organism from being sucked up into your mouth. All right, so that's a common way to collect small organisms that you cannot really touch with your hands. Or if you touch them, they may die or break up, you see. So you use this potters, or putters, you actually call it, to really collect small organisms. You may use bottles, you may use jars, you may use nets, you may use sieves, you may use quadrats. And quadrat, you need to know what a quadrat is. And those are pretty much square or rectangular shape divided into smaller segments or quadrats, you call that. Those segments, those are called different quadrats. You may use line and belt transect as well. Mark, release, and capture. So mark, release, and capture is actually one thing. All right, so you capture something, you mark it, you release it, and you recapture it. All right, and you will see those things mainly on documentaries where those documentaries will be tagging different organisms, and they may tag them with cameras or numbers or different markers, and then they catch them again and then release them. Some of them will also be tagged with trackers to see where they're going and see different movement in terms of migration and all type of different things, or how they feed, how they hunt, and how they reproduce. All right, so there are different reasons why you want to study organisms in the environment. All right. All right, so next thing we'll jump into right now is just a quick look in terms of abiotic versus biotic factors. And so abiotic factors, those are the non-living aspect of the environment. And so those are the physical and also chemical factors as well. So not just the non-living, because non-living include physical and chemical, but not chemical. Physical can also refer to biological. So you have to be careful to differentiate the two. When I say physical, we talk about just the structure of things being there. All right. And so example of that will be including water, the air, the soil, the temperature, the light. Those are the physical nature of the environment. Those are the non-living aspect of the environment as well. And some of them have chemical nature or properties, such as water can also refer to as a chemical. Air can refer to as chemical as well, right? And even the component of soil can also fall under the chemical aspect as well. But generally speaking, and for examination, is the non-living aspect of the environment. And for biotic now, refers to the living aspect of the environment. So different organisms, they are the biotic factor within that environment, all right? So technically now, when you see that, you should think about food chain and food webs, all right, or how organisms relate to each other, whether it be by feeding relationship or simply just a relationship in terms of benefiting one or the other. such as animals and plants, in terms of relating to carbon dioxide versus oxygen, or the exchange of those gases between those organisms. So yes, there are different ways that organisms interact within the environment. Not always feeding relationship, but other relationship as well, including nutrient cycling. All right, so that's a very important thing to look at. All right, so let's start food chain and food web now real quickly. All right, and just dive into some specific information about food chain and food web. All right, so things we're going to discuss right now is energy decrease or energy decrease along the food chain or food web. All right, just to go back one little second, just to kind of point out something here. What to notice difference here is that for food chain, you only have one path for energy to flow, and in a food web, you have many paths for energy to flow. And point to note is that in a food chain, you can, well, both food chain and food web, you see arrows, and the arrows indicating the flow of energy. I know that was on a past paper as well. The arrows indicate the flow of energy or what is eating what. All right, a food web is normally more complex than a food chain as well. Also, difference in a food chain, you will not see omnivores, you can only see herbivores and carnivores, because omnivores eat both plants and animals. Right, you're not going to see that within a food chain, but a food web will show you omnivores. All right, that's very important to note. Also, what is important to note is also if you decrease one organism, what will be the effect on the other organism? For example, if you take out the owl, what will happen to the mouse? And of course, just to let's show you this real quick, what I'm talking about. So let's say I take out the mouse for argument's sake, right? Let's take out the owl for argument's sake. Then there'll be an increase in the mouse. And if there's an increase in the mouse, there'll be a decrease in grasshopper. And if there's a decrease in grasshopper, there should be an increase in the grass. And of course, once you take out one organism, going after that organism or before that organism, you have an alternate change in terms of what is happening. All right? And so again, let me just jump into a specific discussion here that I want to talk about now. We're talking about energy decrease. Again, along a food chain, energy decreases. And you must understand why. Because again, the demand for energy increase, and not only that, the basic reason or the primary reason why energy decrease along the food chain is because each organism is using some of the energy for their own biological functions. And also, what you need to understand again, energy is also lost due to heat as well in a food chain. All right, so that is very important. You have energy lost along the food chain. As you go along the food chain, again, the number of organisms tend to be less. Generally speaking, the number of organisms tend to be less. And the reason for that, of course, because there's a greater demand for energy, and the organisms are normally larger, so therefore they must be less to consume as much energy as possible. All right, again, energy requirement is linked to the size of the organism and how far the organism is in the food chain or food web. The higher you go, the higher the energy requirement due to larger size of organism, generally speaking. All right, and of course, the organism is larger, then they tend to require more energy to sustain themselves and also carry out their life or biological processes. All right, bioaccumulation is very important to discuss as well. And bioaccumulation is when persistent chemicals will remain in a food chain or food web and increase in concentration as you go along the food chain. A reason why chemicals will bioaccumulate is simply because organisms are eating more to sustain themselves and also to maintain their energy requirement, to satisfy that energy requirement. So, for example, a bird may eat one fish for the day, but a man may eat 3 of the small birds for the whole day. So, therefore, you eat more of that organism to sustain yourself because you're larger, and hence the concentration of the poison will increase. We also need to talk about predator-prey relationship. Very important. A predator that will do the hunting, and a prey is being hunted. All right. And we also need to talk about feeding relationship, which I'm going to give you some examples shortly. We also need to know about the importance of decomposers in terms of nutrient cycling, also the decreased wastage in the environment, and also the decreased diseases in the environment. So decomposers are very important. And of course, decomposers will include fungi and also generally or mainly bacteria. So even though these microbes Might sound a little bit disgusting by nature of the name or what we have used to, our perception about them. They're actually useful in recycling nutrients, decreasing diseases, decrease the buildup of waste in the environment, which is very important. All right, so decomposers are absolutely important for us. All right, now some very important special notes right here. We talk about the relationship between organisms. And so some of the relationship that you pay attention to here is parasitism, commensalism, and mutualism. Very, very important relationship that you must know. All right, as you know, parasites, for example, ticks on human beings or on other animals such as dogs, and those are normally tested. All right, commensalism is when, of course, none of them is being harmed really, but only one being benefit, technically benefit in terms of feeding. And mutualism is when both of them are benefiting. All right, so that's very important right there. So parasitism, one being one benefit and next one is being harmed. All right, and then commensalism, one benefit, no harm, and then mutualism, both of them benefit. All right, and so typical example as we talk about, I just jump over that too quick. Lice and mammals, for example, let's talk about dogs and human beings as well. All right, talk about epiphytes on trees, like shelf fungi, for example, you may see them on trees. All right, so the word epi means surface. So epiphytes, they feed on the surface of the bark, the dead bark. Nitrogen fixing bacteria in the roots, nodules of legumes. Those are very important to convert nitrogen into nitrates for the plants as well. All right. Nutrient cycle is also important. So let's talk about carbon cycle a little bit. All right. And carbon cycle, as you know, and I'm just going to show you a diagram, and then, of course, you can add details as we go along. And then, of course, you can also research and reread from the textbooks about these cycles. All right. What is important about the carbon cycle is that photosynthesis is one of the very few processes that remove carbon dioxide from the atmosphere. So trees are very important in maintaining the cycling of carbon dioxide and also to remove carbon dioxide from the atmosphere. Hence, it plays a very important role in terms of reducing global warming. So photosynthesis definitely is an important process. But not only plants will take up carbon dioxide out of the atmosphere, because algae also do that, because once they photosynthesize, they can take out carbon dioxide out of the atmosphere. But photosynthesis is the process, all right, but not only plants. Algae is another organism that will do that. Respiration, both in plants and animals, because plants do respire and plants respire every time, just remember that, all right? But they respire more in the night compared to day, because in the day, photosynthesis rate is greater than respiration. But in the night, respiration rate is higher than photosynthesis. But plants respire all the time, just like all other organisms. All right, so respiration put carbon dioxide back in the atmosphere. All right, as we know that. When all organisms die, they will decompose, and by decomposers feeding on the dead organic materials, they will release some carbon dioxide in the atmosphere as well. When decay takes place and remain in terms of fossil fuel and so on, what will happen is that the burning of a fossil fuel will also release carbon dioxide emission in the atmosphere. And we know those processes. Again, just go over it and spend some time, right? I'm not going to spend too much time here. The other important cycle is the nitrogen cycle, okay? The nitrogen cycle, what is important in terms of nitrogen cycle is that you have nitrogen fixing bacteria in the soil, that is very important, that will remove nitrogen gas from the atmosphere, convert it into ammonia, and that process is called nitrogen fixation. They are fixing the nitrogen gas into ammonia. And then from ammonia, we go into nitrites, which is NO2, and also NO3, which is nitrates. Now, that process is called nitrification, okay? Nitrification, where we convert ammonia or ammonium ions into nitrites, all right? And then we can convert that now into nitrates. And plants use nitrogen in the form of nitrates, okay? And then to send it back in the atmosphere, the nitrates are converted back into nitrogen gas by what we call think about the word denitrification, all right? All right, and so that's how we end up with the different cycling or different aspect of a cycle of nitrogen within the atmosphere. Lightning also could fix nitrogen gas as well into nitrate. So there are two processes here that nitrogen in the atmosphere can convert into nitrates. One is by nitrogen fixing bacteria and lightning. So those are the two processes that convert that. And denitrification is the only process that returns nitrogen gas into the atmosphere. All right. Now, water cycle is very important and very simple. As we know that we have water moving from the earth by directly evaporation or by transpiration from plants as well. And also evaporation can take place when animals, well, like us, when we sweat and the water evaporates from our skin into the atmosphere. Right. So evaporation generally, transpiration takes water into the atmosphere, and then when it reaches the cloud or a certain temperature, then you have condensation taking place. Once condensation occurs enough, then what will happen? We have precipitation taking place. That is rain, hail, sleet, and snow. All right, will take place. And we have runoff water. That is water running off on the surface of the earth or soil. And then we have underground water or groundwater. All right, so next thing we're going to look at here now is the impact of human activities on the environment. Just to give a brief information on these things, what you need to look out for is talk about things like biodegradable stuff versus non-biodegradable and the impact of those things on the environment. So biodegradable are things that can be broken down by biological means, by use of bacteria, for example. So the process of decomposition takes place. And for non-biodegradable, those things will not be degraded or break down in the atmosphere by living organisms such as bacteria. All right, so the only way they break down, you have to break them down physically, either by crushing, grinding, and so on. All right, that's the only way you will reduce them or you recycle them. All right, and of course, also you need to talk about overpopulation as well and overfishing. So these are human impacts on the environment: overfishing, overpopulation. Because the higher the population is the more impact it has on food resources, the use of land, or also the release of carbon dioxide, the use of machinery. So there are a number of things affected by overpopulation, even the spread of diseases as well. Because the closer we are together, is it easier for us to spread diseases among each other. Overfishing. Of course, I understand over harvesting of any organism, as a matter of fact. Because once you over-harvest any organism, you have a possibility of leading to what they call extinction of that organism. Why you have an extinction of that organism? Because the rate of removal is greater than the rate of reproduction. All right? You also have chemical fertilizer and impact of chemical fertilizer. Of course, it could damage soil if you overuse chemical fertilizer. Not only that, if fertilizer get into waterways, then it can cause things like eutrophication, excess growth of algae, as that refers to eutrophication and so on. All right, and of course, you need to know the effects of eutrophication. Products of industrialization, such as, of course, smoke, chemical waste, sewage, and all of those stuff. All right, chemicals seep into the underground water, cause poisoning of water, contamination of water, and so on. Improper garbage disposal, of course, again, can cause habitat for pests, can also increase spread of diseases, can also use up useful lands, all right, and also, of course, it can be unsightly, all right, and of course, can give off bad odors as well. So there are many different things we could have there. And of course, in thing about the ecotourism aspect of it, of course, nobody wants to use beach that is dirty and or nasty. So if you dispose of a garbage in not a proper way, then you realize then of course it may affect tourism or the economy of your country. As well, because of course it will reduce the income from tourism. All right, loss of habitat and species as well, as I referred to earlier in terms of extinction. All right, and if you destroy habitat because you're dumping things in the environment, then organisms may have to migrate because they cannot survive, they can't find food, they're affected by the gases produced, they're affected by the poisons and so on. All right, and of course that can also lead to extinction as well. And also, as I mentioned as I was going along, the impact on human health as well, spread of diseases, right, by accumulation and so on. Right, and even mosquitoes get into dumps and spreading diseases by means of vectors. So you think about malaria, dengue, and so on. All right, typhoid. All right. Now, in terms of section B now, all right, so that's pretty much section A completed. So section B, we're going to talk about cell structures and function. One of the first thing we're going to talk about. Again, it was for you to know the different structures, identify structures and know their functions, such as cell wall, cell membrane, nucleus, cytoplasm, vacuole, mitochondria, ribosome, chloroplast. As you know, cell wall is for general protection. Cell membrane controls what going in and out. Nucleus controls the interactivity of the cell. Cytoplasm is where chemical reactions take place. Vacuole is for storage. Generally, talk about storage of water, waste, all right, as well, and food. All right, mitochondrion is what is producing energy. That's why it's called the powerhouse of the cell, right? Ribosomes that deals with protein synthesis or the formation of protein. And we have chloroplasts where photosynthesis takes place. And also you need to know the difference between animal and plant cells, which I'm going to talk about a little bit later. All right. You also need to know about microbial cells such as bacteria. All right. And structure of bacteria, you need to know that. And specifically notice the nuclei, the cell wall, the capsule, and flagellum, which I'm going to show you these. All right. And of course, protist, an example of protist such as amoeba. All right. You need to know those, the euglena, and so on. All right. Now, a quick difference between plant and animal cell right here. As you know, that cell wall is not in animal cell, but in plants, you need to know that. The vacuole is large and centrally located for plants, but you have many vacuoles within animal cells. All right. The nucleus, generally for animal cell, is technically almost centrally located, but the nucleus is more to one side or one end in a plant cell. There's no chloroplast in animal cell, but chloroplast in plant cell. You need to know that as well. All cells must have cell membrane. All right. So those are some of the basic differences you have to know between plant and animal cell. All right. In terms of bacterium or bacterium cell, please identify for example, the plasmid with a circular plasmid in bacteria. They might have flagellum or may or may not have flagellum. All right, we have the three outer layers that you must know. The first layer, it is called a capsule. They have a cell wall in the middle and then a cell membrane. You must know those three layers. And then we have one circular chromosomal DNA. So the DNA is pretty much circular, one circular chromosome in a bacteria. All right, we have ribosomes that they use to synthesize their protein. All right, and an amoeba, which is a protist, pretty much the basic cell parts, the cell membrane, the cytoplasm, all right, the nucleus, and very important, they have two types of vacuole. Technically, you have a contractile vacuole that will send things in and out, and then we have what they call a food vacuole as well that they use to consume food, all right, and also to synthesize certain products as well. All right, and amoeba, what is important about amoeba? Amoeba can change shape or induce shape to absorb things by endocytosis. All right. All right, so let's go to some very important notes that we need to look at in terms of cells. And so we need to talk about cell specialization, where different cells will carry out specific functions based on where they are located. Their shape and their structures will allow them to do certain things. And so a point to note in terms of tissues, all right, very important, you need to know that different tissues, example, epidermis, you need to know epidermis, you need to know the xylem tissue, the phloem, and xylem and phloem that are transporting tissues, you need to know those, all right. The epidermis, as I mentioned, you need to know also that these are used to form organs as well, all right, and organs include things like leaf and stems, and also roots. Let me add that one in quickly. All right, and also organs now will form organ systems such as transpiration, translocation, the circulatory system, and so on. All right, but these are referring to plant systems that I have right here. All right, and of course, again, you must relate the same type of concept in terms of animals. All right, from cells into the organism. All right. All right, so next movement is going a little bit away from cell. It's still on cell, but this is talking about the transport in and out of cells. And so there are two processes that we need to think about right here is diffusion and osmosis. All right, and you must, and a point to note right here is that diffusion and osmosis, they are what they call passive transport. All right, which means there's no form of energy that is required. Passive transport, no energy required. What is also important is that they will move along a concentration gradient, which means things are moving from a higher concentration to a lower concentration. And so diffusion, for example, is the movement of particles from a region of higher concentration to a lower concentration. until the particles are evenly distributed. Very important. However, now, the difference with osmosis is the movement of water molecules. And water molecules are moving from a higher concentration to a lower concentration across a semi-permeable membrane. Again, what is important is that diffusion and osmosis they require movement from a higher concentration to a lower concentration. In other words, make it very simple. The movement of particles from where there are a lot of that particle or particles to where there's less of that particle. Okay? All right, so let's just demonstrate something real quick. And so active transport now is opposite of passive transport. And in active transport, it requires energy. And energy is required to move substances in active transport. That's why it's called active requirement of energy. So the movement is against a concentration gradient, which means it's moving from a lower concentration to a higher concentration. All right, an example of this is minerals moving into plant roots. So if you think about this, if you have a box and you fill it with stones, for example, and you have to put more stones in it, you have to be stuffing and pushing the stone against the other stones. So you're moving 1 or 2 stones into a box that's already filled with stones. So you're going against the norm, going against where there's less to where there's more. It's kind of hard to do that. So the same thing active transport, they're pushing a small amount of particles to a region where there are more particles. That means you're going against a concentration gradient. Of course, this is an example that occurs in plant roots. Nitrates moving into the plant roots by active transport, because more nitrates is within the plant roots and put more in, it is going against that concentration gradient, moving from where they are low to where there is high amount of nitrates. That is active transport. All right, so please note and know how to explain them. All right, let's dive down a little bit more. Now, the next thing you need to note here is now relating to osmosis is tonicity. And tonicity refers to the comparative or the comparison between concentration in and out of a cell. Or it says comparative concentration in and out of cells. Now, there are 3 types of tonicity. We have hypertonic, we have hypotonic, and we have isotonic. And so for hypertonic, the cell normally shrinks or becomes flaccid. Now, what is happening with hypertonic, look at the first diagram right here, is that outside of the cell will have more of the solute, which is the solution. This solution is more concentrated. So in other words, you have less water outside of the cell and you have more water within the cell. Again, tonicity refers to osmosis, which means the movement of water. So therefore, water going to move from inside. So the blue dots, the small blue dots, those represent water. The green one represent the solute, for example, sugar or salt. So water going to move from the inside to the outside and cause the cell to shrink. In a similar way, hypotonic, water now will go the opposite way. Water moving from outside to the inside. That means the cell is in a dilute solution or a highly concentrated water solution. That means the water is high in that solution. More water than the solute. And so water enters the cell and causes the cell to swell and eventually burst or may burst, especially if using an animal cell or a red blood cell. Now for isotonic, the concentration will be equal. And if the concentration is equal in and out of a cell, technically water going in equals to water coming out. And so therefore there is no net movement of water and the cell remains the exact same. All right, if it was turgid before, it's going to maintain its turgidity. All right, so those are the 3 types of tonicity that we must know. And again, we must also know to compare the difference between animal and plant cell within these type of solutions. And I want to point out the first one, it is hypotonic, difference between animal and plant. In hypotonic, I want you to notice this big difference here, is that the plant cell will not burst, but the animal cell will burst in hypotonic solutions simply because the plant cell has a cell wall that prevents the cell membrane from collapsing or bursting. That's the difference there. Now, in the hypertonic situation, both cells lose water. However, they both become flaccid, but the difference between the plant and animal is that the animal cell become what they call plasmolized, which means the shape will be maintained, but guess what will happen? The cell membrane move away from the cell wall and cause the cell to shrink or become flaccid. All right, so that's the difference there. All right, so the cell membrane does move away from the cell wall because the vacuole decreases in size. All right, so notice the difference in the vacuole in the plant cell, larger vacuole to very small vacuole. So when it gains water in the vacuole, becomes large, increase what they call turgor pressure. So it pushes pressure against the cell wall, okay? And in the hypertonic, it loses water from the vacuole. Cell membrane moves away from the cell wall. The turgor pressure decreases in hypertonic solution for the plant cell. All right. Next thing we'll look at now is feeding relationship on the topic of nutrition. Now, the thing that you must know is autotrophs, cell feeders. We have heterotrophs. Those are organisms that feed on other organisms. And on the heterotrophs, we must know things like saprophytes, parasites. All right, you must know those two at least. So I'm going to show you the different types that we have, all right, that we need to refer to. So autotrophs, you know, they make their own food, generally by photosynthesis or chemiosynthesis. So organisms that use light, as the word suggests, light is photo, so photosynthesis, making food in the presence of light. We have chemosynthesis, those are making food based on absorbing different chemicals from the environment and make their own food. That means there's no need for light, particularly. Now saprotrophic organisms or nutrition refers to organism that feeds on dead plants and dead decaying substances. All right, so we call them saprophytes, okay? Saprophytes. And so saprophytes, example of those will be yeast and mushroom. Notice they'll sit there and just absorb. Parasites, right, or parasitic nutrition, those feed on other living organisms such as tapeworm and lice is a common one to be tested on CSEC examination. All right, we also have our holozoic nutrition, which include us. We ingestion of organic food materials, so we ingest organic food materials. Amoeba also is an example of that as well. All right, so these are different types of feeding. That's how we feed. So generally in the CSEC exam, though, they talk about saprophytes. Even in the last exam, they talk about parasite. No, in the previous exam, and then of course we are always there, and we just refer to ourselves as heterotrophs, really. All right, but the correct term for us is holozoic feeders. All right. Next on our list is leaf structures and functions. All right, and you have to know the external structures of a leaf. You need to know the apex, which is the point of the leaf, the margin, which is outer layer, and then we have the midrib, which is central vein, and then we have our venules, the one that are branching if it's a dicot, and then we have, again, we have veins generally all throughout the leaves. All right, we have the petiole, also called a leaf stem. We have a lamina, otherwise called a leaf blade. All right, and we have a base of the leaf, which is not important because they don't really ask you to label that. Still, all right. Now, internal structures. This is very important. Very, 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 very important. So I find this nice good diagram here online, which indicates the internal structure of a leaf. Really good. All right, and so we must start from the waxy cuticle up top. You must have the upper epidermis. A point to note though, generally you have more stomata on the lower surface than the upper surface, as pointed out. You will have stomata on both sides of the leaves or of leaves, but generally the bottom will have more. I'm going to tell you, well, let me just tell you why. Because why you have more stomata on bottom? Because gases, when they get warmer, they tend to move upwards. So they can easily enter the stomata or enter the leaf through the stomata if the gases are moving upwards. So they capture gases better. You have less on the upper surface because on the upper surface they're exposed to sunlight, and once they're exposed and they're open, then you have a tendency to lose more water. So you have less stomata to prevent water loss or to reduce water loss. All right. We have the palisade mesophyll layer. This is the old layer, right? It's called the palisade mesophyll layer. One of them is called a palisade mesophyll cell. They have the most chloroplast, which means more photosynthesis taking place in the palisade mesophyll cells or palisade mesophyll layer. Then below that, you have a spongy mesophyll cells. They are more spacious. All right, they are more spacious to allow easy diffusion of gases through the entire leaf. A point to note in terms of palisade cells is that they're elongated, so they could capture more light and also contain more chlorophyll, also more chloroplast as well. All right, so they're actually close together as well. That's very important. So they're found on the upper part of the leaf, closer to the surface, to absorb maximum sunlight and contain a lot of chlorophyll to do that. All right. Again, spongy mesophyll, they spread out a little bit to allow easy diffusion of gases. And a point to note that both spongy and palisade mesophyll, as they call it, the mesophyll cells or mesophyll layers, they are the main cells that are responsible for photosynthesis. All right, we have a vascular bundle. And a vascular bundle is made up of a xylem and phloem. Xylem is usually on top, and the phloem is usually in the middle or to the bottom. All right, we have our guard cells, and guard cells, they control the opening of a stomata to allow gases to go in, or also the removal of warm water. All right, so gases exchange takes place at the stomata, just to mention that. All right, photosynthesis. Quick run through of this is that we know that photo means light, and so photosynthesis takes place in the presence of sunlight. And a balanced chemical equation, you must know, and there's a big difference between the word equation and chemical balance equation. And you must know, I'm not even saying you should know, must know the balanced chemical equation for photosynthesis. Never go into a bioexamination without knowing this. So you have carbon dioxide, water, give you glucose and oxygen. There's a 6 in front of everything except the glucose molecule. The word equation, you also need to know the word equation as well. All right? Please make sure you know both. All right. Now, it's very important note here is that one is that the production of oxygen is done by splitting of water molecules by light. So when light split water molecules, they get oxygen. And you also get hydrogen. And the hydrogen is used to combine with carbon dioxide to form glucose. Okay? So again, it said the subsequent reduction of carbon dioxide to carbohydrate. So in photosynthesis, the carbon dioxide turns into carbohydrate, which is glucose or sugar, and that occurs when hydrogen ions react with carbon dioxide, and that comes from water. So in water split, they get two things: oxygen, which is a byproduct or a waste, and some ions, hydrogen ions, and they combine with carbon dioxide. Now, the chloroplast is the side of the chemical reaction for photosynthesis. You need to know that. Chlorophyll absorbs light. All right, and just to make a note is that you need to know the different colors of light. Roy G. Biv, and plants reflect green light, because whatever color you see from something, that's the light is being reflected. All right, and also you need to know what happened to the products, the fate of the products. All right, in terms of photosynthesis. So one is that oxygen is a waste product that is given off, as you know, in the environment. The glucose that remains, it is metabolized to produce energy by respiration. All right, or it is stored in the form of starch. All right, remember that. And also, some of the product can also be used to make things such as cellulose. So the carbohydrates or the sugars that are formed can use to produce energy, can use as storage in the form of starch, and also to make structures such as cell wall in the form of cellulose. And those three things they are all carbohydrates. Sugars that are produced, carbohydrates, monosaccharide. Sucrose that is being transported, that is a disaccharide. Starch and cellulose, those are polysaccharides. All right, so they are all carbohydrates. All right. Next thing on the list now, again, just continuation here. We need to talk about adaptations for photosynthesis. For example, stomata are used for gases exchange. I know that was on the last exam, or if not, well, a recent exam, I should say. And then you have intercellular spaces. Again, we mentioned that in terms of the mesophyll cells, and that for that, diffusion of gases. We also have chloroplasts in the palisade, and they're closer to the epidermis, as I mentioned that earlier, for maximum absorption of light. All right. And then we will take a look now at the factors now affecting photosynthesis, and these are the factors. We have light intensity, concentration of carbon dioxide, water availability, and temperature. And these are the graphs that depict the relationship between the rate of photosynthesis and these variables. So again, notice that these variables are placed on the x-axis because they are what we call independent variable, and the rate of photosynthesis is placed on the y-axis because the rate is the dependent variable. All right, as we know that in low light, so the first one talk about light, in low light, then of course photosynthesis will decrease, as we know why, because light stimulates the opening of the stomata, and if stomata are not open, carbon dioxide can enter. Moreover, light is needed to split water molecules. All right, so less light, less water can be split. And then now for temperature is very important. And notice the graph of temperature that is different from the rest. Temperature goes up to a peak, which we call optimal temperature, then boom, it drops right down. Now what is important here now is that the reason why the rate of photosynthesis increases with temperature is simple this. Because as you increase in temperature, enzymes that are needed for the chemical reaction of photosynthesis will become activated. And once these enzymes are activated, then the chemical reaction can take place. However, after a certain point, the enzymes will become denatured. Simply because enzymes are made of protein and can become destroyed by high heat. Not only that, the stomata will also be closed to conserve the loss of water. All right? So, of course, temperature affects photosynthesis in a positive way, then also increases after a certain point in a very negative way. Carbon dioxide is just like light. I don't even need to explain that. All right? All right. And of course, water is the same thing as carbon dioxide and light. All right? So, three things will look similar. Carbon dioxide, water, and light will look similar, but temperature will look a little bit different because temperature could affect the entire chemical processes and also the enzymes needed. Now, in terms of plant nutrition, we need to know some very important nutrients that plants require, such as nitrogen, symbolized with N. It used to form proteins, and proteins are needed for growth and development. We also have magnesium, and magnesium, which is Mg, needed for chlorophyll production. And also, once you have enough chlorophyll, then it also aids in the absorption of light. We have phosphorus that gives strength to plants. And then we have potassium, and potassium, symbolized by K, is for fruit and flower development. And of course, you will see common fertilizer, which is inorganic fertilizers, called NPK, which means nitrogen, phosphorus, and potassium. And those are very essential nutrients needed by plants. All right. Let's now jump into digestion. So look at the digestive system. Again, I'm just giving the brief notes and different aspects, also give a little bit more details. So when you're reviewing or revising, then at least some of these information pop up back in the head. And so again, it is start out with in the mouth, we have, and what takes place in the mouth, which is physical digestion or mechanical digestion by chewing. All right. We call it chewing. All right, chewing takes place in the mouth by the teeth and tongue. So you have chewing, and the mixing of food also doesn't mention that. All right. All right, and then we have salivary glands in the mouth as well. You don't know what happens in salivary glands. Salivary glands, they produce enzymes, particularly salivary amylase. You don't know that that's starch with the breakdown of starch. So what I want to point out is that in the mouth, you generally have all digestion begins in the mouth by chewing. That's very important, that physical digestion. However, now as you go down, we have esophagus, and esophagus, you have peristalsis taking place when the muscles of this tube-like structure relax and contract to send food down the stomach. And the stomach is where the chemical digestion of protein begins. The pancreas produces different enzymes, trypsin, amylase, all right, and lipase, all right, and of course you need to know what those break down, all right. We have gallbladder. All right, we have a liver that produces bile. Gallbladder will store the bile. We have large intestine. The large intestine releases the production of vitamin D and reabsorption of water, and also to store undigested food in the rectum. And they release that into form of feces. They have small intestine. Small intestine is where all digestion ends. We need to know that, and we need to know what type of food is absorbed. We need to know also the structure of the villi, which I'm going to show you in a short little while. All right, so villus. And this is a beautiful diagram here of a villus. And point out some very important structures that you need to know. is that we have the outer layer, which is called the epithelium. And on the epithelium, we have what we call the microvilli. Those are smaller villi structures that increase surface area or further increase surface area. All right, we have also the epithelial cell. One cell is called epithelial cell. And then inside of the epithelium, we have some special cells that we call them the goblet cells. All right, and those secrete mucus. So sometimes they show you this section as piece that is missing. So the missing part of it shown in an exam is called the goblet cells. All right, and then we have here the blood capillaries, and the blood capillaries made up of what they call the venule and the arteriole. All right, and so the arteriole connect to the arterial end, which leads to an artery, and the venule leads to a vein that they call hepatic portal vein that will carry food substances to the liver. What is also important is that we have the lacteal in the middle. The lacteal is what absorb fatty acids and glycerol. The capillaries will absorb amino acids and glucose. All right, what is not absorbed here? What absorbed in the large intestine? All right, just to make mention of that. All right, so nutrient test, the test for different nutrients. You need to know these basic tests for nutrients. All right, so you need to know about testing for protein, or you test for fat, so you test for sugar as well, so, and also test for starch. I have two different types of fat tests right here, so let's pay attention to them. So let's go with the starch test first, in the first row. So starch test is used, you use the reagent of iodine solution to test for starch. Iodine itself is yellowish brown, and iodine changes to blue-black if starch is present. So again, it is not the food that is changing color, but the solution changing color. So please do not confuse that. When you test for proteins, they use burette solution, and you change the color. The color is blue. Burette is a blue solution, and it changes to purple if protein is present. There are two common tests for fats, and so one is using ethanol and water. Both ethanol and water, they are colorless, but the mixture becomes cloudy if fat is present. And then there's the next one is called a grease spot test, or the paper test, which you just rub the food substance on a piece of grease paper, on a piece of grease, a piece of paper really. And if you see a grease spot, they know that yes, fat is present. All right, so do not use a particular reagent, just use a piece of paper. The reducing sugar test, use Bendix solution, and Bendix solution is also blue, just like the burette solution. And if sugar is present, then you get a red, brown, or orange color. Of course, more like the orangey, yellowish color as well. That is also a positive test. So anything that is reddish, brownish, orange in color, yes, it shows that reducing sugar is present. All right, and a matter of fact, for the Bendix solution, just to make mention to that, you have to use heat to warm up the mixture so the reaction can take place. All right, enzymes. Let's talk about enzymes a little bit. Now, enzymes, now they're called biological catalysts. Why they're called biological catalysts? One is because they work in living organisms, and they're also made up of proteins. All right, because protein is a biomolecule. All right, and we have catalysts. They are called catalysts because they alter chemical reactions, and so they are called biological catalysts because they alter chemical reactions in living organisms. So that's why they're called biological catalysts. All right, so some specific properties of enzymes that you must know is that they're all made up of proteins. All right, they have the low activation energy, so chemical reaction can take place. They are substrate specific, which means they only act upon certain food or certain nutrients or certain substances. All right, so they are substrate specific. So in other words, whatever break down protein will not break down starch. All right, they break down specific things. They are reusable because they themselves are not used up in the chemical reaction. They remain unchanged. So they are reusable. They are sensitive to pH, so they are pH sensitive. They're denatured by high heat as well. They are inhibited by poison, so poison will stop them from working or prevent them from working effectively. All right? So those are some of the things that you need to know about enzymes and the properties of enzymes. Now, the respiratory system and also gas exchange is something that you need to pay attention to. You need to know your different structures, such as the nasal cavity, pharynx, which is your throat, the larynx, which is the windpipe, your trachea, and your trachea is made up of rings of cartilage to prevent it from collapsing when you're swallowing food. You need to know that. We have our bronchi. One of them is called a bronchus, and those are the subdivision from the trachea going into the what they call a bronchiole, so those lead directly into the lungs. And then we have the bronchioles. Those are the subdivision of the bronchi. And at the ends of bronchioles, we have the alveoli. Those are small sac-like structures. And below the lungs, we have a muscle called the diaphragm. And that's very important in breathing. Helps in breathing. Very, very important. All right, next thing on the agenda is we're going to talk about now is gaseous exchange, real quick. So this is the structure of the alveolus. All right, as you see right here. Gas going in, gas coming out. So you have inhalation, the gas from inhalation coming in, and the gas going out is what's going to be exhaled. And so around the alveolus, we have capillaries, blood capillaries that carry blood cells. And particularly, we have red blood cells that coming in that is deoxygenated, blood coming in, and what going out is oxygenated blood. So the purpose of the alveoli is to carry out gaseous exchange, remove carbon dioxide from the blood, and replace it with oxygen. That's what they call gaseous exchange right there. So what is important about the structure of an alveolus to make it effective? It has a large surface area for gaseous exchange and effective gaseous exchange. The walls must be thin so diffusion can take place easy. Must be moist. I have moist twice. Must be moist. And also great supply of blood capillaries. All right, so ignore one of the moist. So it's moist, thin, large surface area. All right, and supply of many blood capillaries. All right, and of course you know why, because the blood capillaries will carry the carbon dioxide towards them and take in the oxygen that they are given. All right, so the gaseous exchange takes place there. All right, what you also need to know is the difference between aerobic and anaerobic respiration. You need to know that. And to be honest with you, I have most of these lessons posted on the biology videos or biology videos or topics. So you can look for that playlist on my channel. You will see a lot of different videos on most of these topics. All right, I don't reach all yet, but eventually. All right. For aerobic respiration, it requires oxygen, occurs in the mitochondria, produces more energy or ATP, takes a longer time to occur because you have to have a complete breakdown of glucose. So that's a big thing. I should put that one there. So aerobic is a complete breaking down of glucose. What is produced for aerobic respiration? The waste are easier to remove. For anaerobic respiration, no oxygen is required, occurs in the cytoplasm. Please make a note of that. Produces less energy or ATP. It is faster because it results in an incomplete breaking down of glucose. No production of water generally. The waste are harder to be removed as well, such as lactic acid and also alcohol. All right. Now, these are the three examples of the balanced chemical equations for the three different types of respiration you need to know. One is lactic acid fermentation and alcoholic fermentation. Those are the two anaerobic respiration. And if you notice, you need to know the formula for lactic acid, which is technically half of that of glucose. Remember, half of glucose, the exact half of glucose is a lactic acid. All right, and so you just put a 2 in front of that to balance it up. All right, and you get ATP out, you get about 120 kilojoules of energy. For alcoholic fermentation, you get a little bit more energy than lactic acid. Alcohol is produced, which is ethanol. You get carbon dioxide, and if you're baking bread by using yeast, then definitely you get carbon dioxide to make the bread rise. And the amount of energy you get out of that is 210 kilojoules of energy. Now you have aerobic respiration that we need to know by heart, and that produces 2,880 kilojoules of energy. All right, and you notice the value of ATP respectively. In anaerobic respiration, you only get like 2 ATP, a total of 36 ATP from aerobic respiration. So you get much more energy. All right. Now let's talk about transporting organisms. And just to give a general idea what it is the movement of substances within organisms. And we need to know about the transportation of oxygen, minerals, amino acids, carbon dioxide, nutrients, sucrose, for example, in plants, just to make mention of that, hormones, glucose, and also waste. We need to know the movement of these substances or the transportation of these substances within different organisms. All right, let's talk about blood cells a little bit. Now, there are 4 blood cells that we need to particularly pay attention to. All right, 3 different types generally, but 4 specific ones. You have the red blood cells, which are called erythrocytes. You have lymphocytes, which is a form of a leukocyte. And we have phagocyte, which is another form of a leukocyte. All right. I make a mistake right here. I'm gonna cross this out because it's not a. Oh, I put this here. This is called a thrombocyte. This is called platelets. So I should have platelets right here. Okay. So the last one is platelets, is a thrombocyte. So this is platelets. All right. So platelets. I don't know how I made that mistake. Platelets. All right. So that's a platelet right there. And platelet is definitely a thrombocyte. Okay. It deals with blood clotting. All right. Leukocytes, which are these two right here, they are used in immunity to protect you against infections. And red blood cells, they are generally for transportation. All right. All right. So let's jump into the next aspect right here now. Talk about blood vessels now. So there are 3 blood vessels you need to know: arteries, veins, and capillaries. Now, arteries compared to veins, they are thicker walls. The lumen, which is the cross section right here, is narrow compared to veins. Veins have larger lumen. All right. Also, veins have valves. Arteries do not have valves. Capillaries, they are 1 cell thick for easy diffusion of substances to cells and from cells into them. All right. That's very important. Capillaries also connect arteries to veins. All right. Very important. And blood flow. Blood is always flowing from arteries to veins towards veins. All right. So just please note the difference between arteries, veins, and capillaries. Now, in terms of the heart, you need to know the structure of the heart, the chambers of the heart. You need to know the different blood vessels that some of you already know. Know the direction of blood flow in and out of the heart. You need to know that as well. All right. You need to know and go through that. I have also circulation on my channel. You can view that if you desire to do so, if you forget certain basic stuff. And I will explain in terms of the different chambers, what is going where, and an easy way to remember how blood is flowing. No matter of fact, I'm just write it down. So I will put in that as well as a link. All right. So I'm gonna put some link in the description that you can watch specific lessons. Not all the lessons that I have. Some of them you have to search on your own if you do want them. But I have most of the lessons in video forms or different lessons. All right. Now, what I'm going to talk about right here now is difference between 2 important type of pressure that you may experience in your body, and that is called systolic and diastolic. And just to make a note that normal blood pressure is 120 over 80, 120 over 80. 120 refers to the systolic pressure, which means blood is moving from the heart into the body. So that's what I call systolic, moving to the systems of the body. And so that is when the heart is being emptied with blood, pushing out. So the heart is contracting, in other words. All right. And for diastolic now is when the heart is being filled with blood. So blood going towards the heart and into the heart. The heart being filled with blood, ready to go through systolic. So it fill with blood first, then push out. Fill, push out. That's the heart keep beating. Contracting and relaxing to make these 2 processes work. So they must work together. All right. Now, in terms of immunity, again, I'm not going to go through everything, but there's some specific things you need to know about natural immunity versus artificial immunity. All right, natural immunity, of course, that is you acquiring that in terms of exposure directly to the pathogen. You know, think about when you're babies and growing up, you know what I mean? You have also passive, I think I missed out one. Passive, you get it from your parents. And then artificial, you get it through vaccines. So when you develop on your own, which is natural immunity, you know, you're exposed to the things in a natural environment and you develop that naturally on your own. Artificial is the one that you take vaccines for. And then you have passive is the one that you get from your mommy. All right. You think about phagocytes and their role in immunity, lymphocytes as well. Phagocytes will engulf, as you know, engulf and digest. Lymphocyte will produce antibodies. And antibodies, those are chemicals that will destroy what they call antigens. Antigens are produced by microbes. So bacteria, viruses will produce antigens, and those are specific proteins that will be produced by these microorganisms that can destroy your body cells. And so antibodies will attack antigen and destroy them. Pathogens now. Pathogens, those are disease-causing organisms, such as bacteria, protists, and so on. Vaccines are what are developed to produce or to trigger immunity. And vaccines back in the days are normally a dead form or an inactive form of the disease itself. So your body could quickly develop antibodies against it, and just in case the real thing comes, then it is ready and prepared to tackle that. Then we have transmission of diseases between persons or between other organisms, such as mosquito, which are vectors, and human beings. We have STDs or STIs. All right, we need to notice those and also pay attention to different specific ones. So please go through the list and know them. All right, root year cell. Root year cell, need to know about root year cell. Root year cell is very important in terms of increasing surface area for roots to absorb water and mineral salt. Root year cell have large vacuole. Just like a regular cell, there will be mitochondria, there will be cytoplasm, there will be nucleus, there will be cell wall, there will be cell membrane, just like a regular cell. All right. Now, adaptations of root year cell is that root year cells, again, as I mentioned, they have thin walls for easy diffusion, just like any absorbing structures in living organisms. They're normally thin. They have large surface area for effective absorption. They have large vacuole to store large quantities of water. Root year cells have many mitochondria to provide energy for active transport. And remember, what moves into the roots by active transport is mineral salts. Water will enter by osmosis. All right, but mineral salts, nutrients, nitrates will enter by active transport. They contain transporting proteins to take in specific minerals or mineral ions, again mentioned like nitrates, all right, potassium, and so on. All right, transportation in plants particularly, we need to focus on evaporation of water from the uppermost part of the plant. That's transpiration as a transportation. So transpiration is removal of water from the upper part of the plants, particularly the leaves. Very important to note, and water will escape through what they call the stomata, just to mention that. So water will go from the root all the way up. All right, so what is important to know about water properties, that water is cohesive and adhesive. And so because water is cohesive, which means it can stick to itself, and water is adhesive, meaning it stick to other things. And because of this property, water can easily pass through the xylem. Again, we need to talk about capillary action. And capillary action is the movement of substances through narrow tubes. So water can move through the narrow tubes of a xylem easily, climb up the tubes. That's what they call capillary action. We need to talk about transpiration pull. And transpiration pull occurs when water evaporates from the leaf or from the plant, and because water is cohesive and they interlock to each other, that means as some particles are evaporating, it will pull other particles behind it, right? So that's what transpiration pull occurs. Is a pull or a suction caused as water evaporate from the plant and cause other water molecules to pull along with it. What is important about transpiration pull is simply the warmer water in the leaf will be replaced by cooler water from the soil. So it's very important to cool the plant as well and make cooler water available for photosynthesis. All right? Now, factors affecting transpiration, we have temperature, we have wind speed or air movement, we have light intensity, we have humidity. So for temperature, the higher temperature is the more kinetic energy the particles will get, and so the more movement and evaporation and also the faster diffusion. I need to know these very important notes right here. Wind speed, the faster the wind is the more water that will move away from the plant, and this creates and maintains a steep concentration gradient for diffusion. Also, we have light stimulate the opening of the stomata. That's for light intensity. So you have more light, meaning stomata will be open. When stomata are open, there's a higher potential for diffusion of water. For humidity, which is opposite in terms of the rate. So for high humidity, you have more water, which is more water in the atmosphere. And if you have more water in the atmosphere, then you have more water being retained in the plant, because guess what? You have lower humidity, create a steeper concentration gradient for more diffusion. So high humidity, less transpiration. All right? More humidity, less transpiration. Decrease in humidity, more transpiration. So they're opposite. So they are actually inverse of each other. So the higher the humidity is, the lesser the transpiration will be, or the less the water loss will be. Because it's harder for you to put water in the atmosphere that's already filled with water. All right? So that one, the graph will be different for that one for sure. All right? Now let's talk about excretion. And what is important in terms of excretion is removal of waste from the body, but importantly, not just any waste, but metabolic waste. That's very important to note. So removal of metabolic waste from the body. All right. So let's just talk about it a little bit further now. Let's talk about the structure of the kidneys real quick. When you know the structure of the kidneys, I'm going to point out things you must know. You must know about the cortex. You have to know that. You have to know about the medulla and the pyramids as well. So the region right here where the pyramids are, that's what they call the medulla, like in the middle section of the kidney, okay? And then we also have our renal artery, renal vein. But quite frankly, you'll not see colors on the exam. So what you're going to look out for is arrows. Arteries will go to. So if you see the arrow going to, that will be the artery, and the vein will come in from. So you need to know that, okay? So arteries go in, veins come from. All right, and what coming from the kidney, the tubules that come from the kidney is what they call a ureter, that is carrying urine to the urinary bladder. All right? All right, the nephron. So the nephron is the filtering unit of the kidney, you need to know that. And the nephron is in two parts. Part is in the cortex and part is in the medulla, you need to know that as well. So the uppermost part of the kidney right there is in the cortex, the lower part is in the medulla. So the loop of Henle is in the medulla. The Boseman capsule is in the cortex, all right? So you definitely need to know that as well. And so the nephron now is a substance from the blood that are filtered into the what they call the glomerulus right here. So the glomerulus is where filtration takes place. Motherfucker, it's called ultrafiltration. All right, because filtration occurs under high pressure, so it's called ultrafiltration. We have our tubules of a kidney. So we have loop of Henle at the bottom right here. We have our convolution. We have a proximal and we have a distal convolution. We have a collecting duct and so on. And specific, pay attention to this notation right here. All right, that we have a lot of absorption taking place at the convoluted areas or the part that actually curve or bend. And then we have some substance absorbing the loop of Henle as well into the capillaries. Notice the capillaries are covered around it. And then by the time you reach to the collecting duct, that's where urine is actually formed. And urine is a mixture of salt, water, and urea. All right, so skeleton system at this point. All right, let's talk about that. So what you need to know about the skeleton system, you need to know about bicep and triceps. You need to talk about antagonistic muscles. You need to talk about types of muscles, skeleton muscles, smooth muscles, and cardiac muscles. Notice, know the difference between them and to identify them. You need to talk about bones. Minerals needed for bone formation, such as calcium. You need to talk about ligaments that connect bone to bone, and tendons that connect muscle to bone. You need to know to identify those. You need to talk about diseases that affect the bone, such as rickets, which is lack of vitamin D, and also calcium. And of course, phosphorus to a lesser degree. All right. So I'm going to look at some specific parts of the skeleton system right now. So generally, you need to know different bones. I would say notice things like the skull or what is called cranium. You need to know the clavicle, the sternum. So the major one, humerus, you need to know that. Radius and ulna. All right. Tibia, fibula. All right. The patellage is the kneecap. You need to know those. The femur, you need to know those. The pelvic bone, you need to know those type of stuff. All right, the vertebral column. So those are the very basic bones that you need to know. You know what I mean? But very specifically, this is what normally tested, or generally you see most times and most bio exam, is the bicep and tricep muscles working together. Again, bicep is in front, the tricep is at the back. And notice the tendon connecting the muscle to the bone. So those are things you need to identify there. And let me go back 1 second, 1 quick second here. All right. So in terms of the bone, just to make mention of these muscles and bone at the same time, is that if the arm is being, you're going to flex the arm or pull the arm up, notice what happened to the bicep. The bicep is contracting and the tricep is relaxing. The opposite happen here. If you extend the arm, then the bicep will relax and the tricep will contract, get shorter. All right. And they work oppositely, what they call antagonistically to get to each other, to bring about movement. So they are antagonistic muscles, all right? Now in terms of irritability, just to make reference to this, that you need to focus on in terms of irritability, you think about tropism and taxism. I have videos on those as well. You think about nervous system, brain, and reflex I'm going to look at that. I'm going to talk about stimulus, which is any change in the environment that triggers a response in the body. Again, you need to look at the description for links, for tropism and taxism. For sure, I'm going to give you that. All right, the nervous system I'm going to run through real quick. Even though I have videos on that, you can also run over to that as well if you want. All right, we want to talk about types of neurons. We want to look at label diagrams of the brain and the reflex arc. So I have those coming up just now. All right, so the brain. The three major regions of the brain that you must know is the cerebrum. We need to know the cerebellum. We need to know the medulla oblongata, which is also called the brain stem. And then connected to the brain, we have the spinal cord. So the spinal cord is not a part of the brain, but the brain and the spinal cord make up what they call the central nervous system. The peripheral nervous system is all of the nerves, which include the cranial nerves, the spinal nerves. So the cranial nerves connect to the cranium or the brain. Well, not as a cranium, to the brain in the cranium. And the spinal nerves connect to the spinal cord. All right, so the cerebrum controls voluntary reactions and memory, example, emotions and intelligence. You're walking and talking and so on. Your cerebellum controls balance and coordination. Your medulla oblongata, I do the spinal cord first. The spinal cord controls reflex action, and the medulla oblongata now controls involuntary reactions, such as the heartbeat and breathing and so on. All right, the neuron. Now, you need to know a neuron, and you need to know different parts of a neuron, such as the dendrites. The dendrites is like the leaf, branchy part of it. The most branchy part, you have a cell body, and in the cell body, you have a nucleus, like any other new cell. You have our swan cell that make up part of the axon. And what cover the swan cell or the axon is what they call myelin sheath. That's for insulation and to speed up the mode of transmission of impulses. You also have between two swan cells, you have the node of Ranvier. All right. We also have axon terminal, or what they call nerve endings. All right. And what to note also is that impulse will travel from the dendrites always. The dendrites what pick up impulse and transmit it towards the nerve endings. So impulse always enter through the dendrites. Always. Never forget that. Okay, so no matter how the nerves turn and shape, the dendrites is what picking up the impulse first and foremost. All right. All right, next we'll look at now is the type of neurons. And so we have three basic types of neurons right here. We have a sensory neuron. Easy way to know the sensory neuron, the cell body is at the side. Side, S. Sensory neuron, cell body at the side. The sensory neuron, it transmits impulse from the sense organ to the brain or the spinal cord. So pretty much once it's run from the sense organ, that's the sensory neuron. The relay neuron now, it's in between both sensory and motor neuron. A matter of fact, if you look at the reflex arc, you see the relay neuron within the spinal cord. All right? In the middle of the gray matter, matter of fact. All right? So look at it within the gray matter. And then now the motor neuron transmit impulse from the spinal cord to the effectors in the muscles. All right? All right, so we want to jump now to the reflex arc. Can I bring this point out? And so you need to know the direction of transmission of impulses. I must put some arrow heads to kind of give you what I'm saying right here. So from our sense organ, we have some small cells in that called receptor cells. So the receptor cells pick up the impulse, all right, or the change in a stimulus. And then now what will happen here, send it along the sensory neuron. How we know the sensory neuron? There's a cell body at the side again, see it right here. And the cell body is usually situated in the root of ganglion, all right? So in the root of ganglion, that's where you find typically the cell body on the sensory neuron. And then we have a relay neuron within the gray matter that connects both the sensory neuron and the motor neuron. And then the motor neuron leads to the muscles. All right. And of course, you have a gray matter. And the central point within the gray matter right here, we call it central canal. And then we have a gray matter and the white matter make up the spinal cord. All right. All right, so again, you need to know just how it works. Now let's look at sense organ real quick. I'm not going to spend too much time on them. All right, so let's go to the ear, and please, you need to know the different structures. You need to know, especially, let me just point out the aerosicles, malleus incus stapes, or otherwise called hammer, anvil, stirrup. Those will vibrate and amplify the sound as it going along. The semicircular canal is for balance. All right. And then we have the cochlea. The cochlea will convert the vibration into impulse, and then that will travel to the auditory nerve, to the brain. All right. And of course, we have our round window and oval window. And our round window, they will help in terms of maintaining pressure along with the eustachian tube within the ear as well. All right. In terms of skin, I must kind of refer that. Again, just please go through the diagrams and make sure you know the different parts, the different layers, epidermis, dermis, and we have the hypodermis. And then different structures that I talk about, the sebaceous gland, which is the oil gland. We have our erector pili muscle. I think that was on the last exam. And again, please need to note the skin function in terms of temperature control, which is a part of homeostasis. And we may sweat to reduce heat. We also have vasodilation versus vasoconstriction. Vasodilation is when you're hot or the environment temperature is high. And then vasoconstriction now is when it is cold, you retain heat by constricting the blood vessels and moving away from the surface of the skin to retain heat in the body. You also need to know the skin helps us to protect us from UV or ultraviolet radiation, also protect us from pathogen as well and physical damages. What is also important is the human eye, to pay attention to that, different parts as well. You need to know the different structures of human eye, the choroid, the retina, the sclera, the functions of those parts. The fovea, which is also called the yellow spot as well, where you get the most vision. All right, did you talk about rods and cones? All right, that are the sensory cells that are within the retina that will detect light. All right, cones, they are for color vision. Rods are normal for black and white. All right, you have a lens. You need to know how the lens are adjusted as well by the suspensory ligament and so on. You need to know about the iris. The iris controls the size of the pupil. You need to know that the cornea as well. Vitreous humor and aqueous humor, you need to know those as well. All right, so please make sure you know the different functions of the structure of the eye. What is important is just a quick look at what happened to the eye in terms of in dark and dim light. So in the bright light now, well, bright light and dim light, I should say. So in bright light, what happened to the eye is that the pupil itself would constrict. Why the pupil constrict is to prevent less light from going into the eye. And how this occurs is because the radial muscle, they relax, while the circular muscle, they contract. The opposite is true in dim light. In dim light, the pupil dilates or get larger. And how that occurs is because the radial muscle, they contract, while the circular muscle, they relax. So you need to know the difference in dim light versus bright light and the reason why. All right. Now, other important topics you need to pay attention to is reproduction in flower, sexual reproduction, and menstrual cycle. I have all these videos posted on the bio playlist. You can check those out. I also have pollination and fertilization as well. Diseases, you need to pay attention to diseases, deficiency diseases, STDs, and so on. So deficiency diseases such as anemia, you need to know that as well. Rickets, which I mentioned earlier, I need to pay attention to those. Lack of vitamin C, talk about scurvy and so on. You need to talk about infectious diseases, non-infectious diseases, contagious diseases, and non-contagious diseases. You need to know those as well. They're very important. They normal test on those things. You need to know things like malaria, dengue, and all of those. All right? Very important. All right, so we're not finished yet because we have a number of things we want to look at. We want to jump to section C now, which I covered not everything because I don't do evolution and natural selection, but I'm going to talk about that a little bit. But what is important now in terms of genes and genetics, so you need to know this, is that genes are segments of DNA that carry coded information or instructions for our individual characteristics or traits. And from parents to offspring, genes are responsible to transmit those characteristics. Very important. Now, some very important terms we need to know right here. We need to talk about alleles. Alleles are variant form of genes. We need to know that. Alleles can be dominant or recessive. For example, if you use uppercase R, it's for red, and lowercase r is for white. Notice we use R's, even though we're looking at two different colors, because we're looking at the same trait, which is color. All right? So uppercase is for dominant, and lowercase is for recessive. Just to make a note of that. All right? Genotype, just to mention genotype. So genotype now is the genetic makeup of an organism that determines the possibility or possibilities of its phenotype. So it's a combination of alleles that determine your phenotype. So in other words, the genetic makeup of the organism. Now, the different types of genotype that we may have homozygous dominant, which means two uppercase letters, don't have to be hard. We can have heterozygous, and hetero means different, homo means same, so two same, but if it's dominant, it must be uppercase. And if it's heterozygous, mean uppercase and lowercase, so hetero means different. And then we have homozygous recessive, and homo means same, recessive means lowercase, so that's why we have this, and that's a combination. Notice the difference between alleles and genotype. Alleles, one letter. Genotype, two letters. All right, so let's jump to an example. So again, we're going to use this now and then we'll go into the example shortly after this one. All right, let's go through this first. So phenotype is a physical or the observable traits or features of an organism. So they can notice or see about an organism that we call phenotype, physical nature of. All right? And then now, as an example now, let's say we have alleles. Red, uppercase R is a red, lowercase r is a white. So the possible genotype that we may have here is, if we have two uppercase R, that means this must be red, because the dominant allele will always show the characteristic, okay? So if we have uppercase and lowercase, which is heterozygous, it will still be red because again it has an uppercase R, and two small r's will be white, again because there's no uppercase R to show you red. Okay, so recessive trait can only seen in the homozygous form. All right. So once you have an uppercase letter, the recessive characteristic will not show. All right, so a Punnett square nose is a box to show the result of a combination or a cross between two genotypes or two different parents. All right, and so the first thing I'm going to do in terms of working out any genetic question is first you have to determine the alleles, then look at the genotype, look at the gametes that are formed, then look at the cross, and then look at the possibility. So this is the order which you do Punnett squares. All right, so let's say, for example, in flower, red petals are dominant over white petals, right? A heterozygous flower was crossed with a white flower. This is the only information given. The fact that say red is dominant over white, it tells you something. So we know that red, this is allele right here. So red, we know that red is dominant over, so we give red uppercase R. White must be lowercase r, simply because red is dominant over white. That's the information given. Now the second thing is said that we have no, well, let's put this back quickly and then we go to the second thing. So red again, uppercase R is red, lowercase r is white. So based on the information given, we say one of the parents is heterozygous. And if you're heterozygous, which means you have an upper and a lower. But the thing is that this one will be red. So this is a genotype. So this has to be what? Red. And it crosses a white flower. And if you're going to be white, you can't have an uppercase R. You must only have lowercase r's. All right? So these are the two parental genotypes. Now the gametes now is how we separate the genotypes. So when we separate the genotypes, for example, the heterozygous is an uppercase R, lowercase r. Those are the possible gametes that we have. And for the white flower, we can only have small r gametes. Now, once we put them in, we're going to put these now in the Punnett square. Put them in the Punnett square, we have something like this. From the heterozygous flower, from the white flower on the side, so the white flower is on the side, and the heterozygous flower is on the top. So the top part for the heterozygous, and on the side is a white flower. There's no rule to say you must put one on the top, one on the side. There's no rule. You can put any one there. The results will be the same. Now, by crossing in this box is row by column. So you have uppercase R, lowercase r. This one, row by column, two lowercase r's, row and column. This box right here matching up column and row, again, uppercase, lowercase r. So even if a lowercase r come before the uppercase r, a rule of decency, we just try to put uppercase before the lowercase, right? And then the last one now is two lowercase r's. All right. Now the genotype possibilities here, if you notice, we only have 50%, two of this and two of this. So 50% chance of getting these genotypes. So 50% of getting heterozygous dominant and 50% chance of getting homozygous recessive. And then now for phenotype possibilities, we know that these will be red, these will be white. So 50% chance of getting red and 50% chance of getting white. All right. Now let's do sex-linked inheritance real quick, and then we can wrap up real fast, because this is pretty much on the last. So this is a quick run through. I don't want to cluster too much with too much information. This also kind of bring back some little points that you may miss in the regular classes. Now for sex-linked inheritance, very important information they must know. Generally speaking, though, CXC mainly focuses on X-linked traits. Mainly focus. Most of the questions that we have seen in the past, they are X-linked. All right, but point to note though is that fathers can pass on X-linked alleles to their daughters, but not to their sons. And you know why? Because guess what? Sons will get their X chromosomes only from their mother. Okay? And then the third point to note here is that mother can pass sex-linked alleles to both sons and daughters. Because the son can get the X, the son has to get the X chromosome from the mother. The daughter now can get the X chromosome from either mother or father. All right? And that is why in any case of a recessive X-linked, a recessive X-linked trait is mainly affected, males are normally affected by it because it is more likely to be transferred to the male from the mother. Now, the fourth point you need to note here is that only males are affected by traits linked to the Y chromosomes. Okay, so if it's a Y link, only males are affected. All right. The next thing we need to look at right here, very important, 2 points are important right here. For recessive X-linked traits, a female will express the phenotype only if she is homozygous recessive. Only if she is homozygous recessive. However, now, if a male is recessive, the recessive allele from his mother, if a male receives the recessive allele from his mother, he will express that phenotype no matter what. Because a matter of fact, he will only have one X chromosome anyway. So if you get that one recessive chromosome from his mother, he's going to show it up. And for this same reason, males are more likely affected by these X-linked chromosome traits. For example, color blindness, for example. All right, so to understand this a little bit further, this is a mother right here, this is a father. Mother with X chromosome, father with X and Y chromosomes, all right? So first thing, let me just go by piece. Look at the colors of these lines. So let's say the first daughter get X chromosome from the mother, this striped X chromosome, and this full dark X chromosome from his father, then she's going to be this, okay? So you notice that now, if the X-linked, it can be affected by mother or father, okay? So if a mother is carrying the recessive allele and the father is carrying the recessive allele, then definitely this daughter will be affected, okay? Next now, look at this now. A mother given this chromosome and a father given this X chromosome, that's the next possibility of a daughter. So notice there are two ways a daughter can become. One, with one type of X chromosome from mother, or the next X chromosome from mother. But the daughter will always, notice the two daughters will always have the father X chromosome. But the other X chromosome in the daughter could be either or X chromosome from the mother. So notice it. So if a father is carrying that X chromosome, then there's a light chance of the daughter being affected, especially if the mother is also affected. Now for a son can get this X chromosome from the mother, this Y chromosome from the father. Also, next possibility here, he could get this X chromosome and also this Y chromosome. So the son is always getting the Y chromosome from the father. All right. Now, let's look at this example here now. This is inheritance for hemophilia, and this was given on one of the previous exams, I think, yeah, since recently. So this is a father right here, X and Y chromosome, and this is without hemophilia, so it's normal. So notice the color pink is for the hemophilia. All right, and hemophilia, of course, is the inability to clot the blood. If the mother now is a carrier, that means she's a carrier. She don't show the traits, but she have it hidden because it's recessive. Okay. Now this is a son being born with the father Y chromosome and the mother unaffected hemophilic X. So he is unaffected. He's normal. However, if the daughter gets this X chromosome, which is normal from the father, and the affected X chromosome from the mother, she becomes a carrier. If the son gets this, which is a pink chromosome, which is a pink X1, is the affected chromosome from his mother, he has to be hemophilic because the Y chromosome has no effect on the X, you see? The next daughter could get a two dark chromosome from the mother and father, and so therefore she'll be unaffected. And she's not a carrier, she's just normal, you see? So you have a daughter could become a carrier, a son be affected, or a son be normal. All right, so that's a possibility right there. All right, so genetic variation now is the next thing we need to look at. I'm going through this real quick. Genetic variation is the difference between individuals of populations. All right, and so we're looking at two different types of variation, continuous and discontinuous. All right, and so in continuous and discontinuous, what's the big difference here is that continuous influenced by genes. Genes influence the continuous, and also the environment influence the continuous. But discontinuous is influenced by genes only. Only genes are affected by the discontinuous. Environment has nothing to do with, nothing on discontinuous variation. All right? And so by the examples you will see. Now, for continuous variation, it's on a continuum. It's a gradual change over a range of time. The majority of individuals are usually in the middle. All right, so you have an average weight, for example, average height. Yes, so most people fall into that. All right, again, just to make a note that continuous is transmitted by genes or affected by genes and also influenced by the environment. Now, for continuous variation, as just to make a note or an example of this, for example, you have different traits, for example, height, and of course, the majority will be in the average. So definitely it's an example of height in a population. All right, so you look at something like that, a graph looks something just like that. All right, in terms of examples now of continuous, we have here, all right, just hold on, example coming height. We have height, that's a continuous variation. Notice there's not either or either, so it's at various ranges of height. All right, our next example of continuous variation here is weight. We have different weight as well among a population. People may have different weight, so there's no one weight for any given person. All right, there's no different category. Shoe size is an example of continuous. So some person have larger feet than others, for sure. All right, we also have discontinuous variation now. Discontinuous variation now is either either. So there's a distinct categories, definitely. No intermediate, no in between, no average, nothing. All right, it's either either. It is not influenced by the environment. It's basically only solely based upon genes or chromosomes. All right, so only genes affect the discontinuous. And so examples of this, we're going to look at them real quick. All right, and again, if you look at the graph situation, you will only have two categories, either group 1 or group 2 or group A, group B, whichever the case may be. And now for example of discontinuous, as you all know, the first one I'm going to talk about is your gender, either your boy or your girl. You fall in either of those categories. There's no in between, or no maybe so. All right, boy or girl. All right, your fingerprint is specific to you. There's no categories for fingerprint that are discontinuous. You're born with it, you die with it. Tongue rolling, either can roll the tongue or not. All right, that's another discontinuous variation right there. So we have different examples of both of them. Just so can make leaf size, for example, in plants, length of leaf, chunk size in elephant, for example. Speed, definitely. The length of a hair, the blood pressure. Yep, those are continuous things. Discontinuous, maybe the color of petals. Color of petals don't really change. Texture of seed, coat, definitely high color and so on. Those really don't change. Blood group don't change. Your earlobe, whether it be attached or detached or disattached, those things don't really change. All right. So this is example of an earlobe right there, attached or detached, if you notice that. Factors that affect variation may include the diet, mutation, migration, genetic drift, random mating, physical activity. So these things can change or you appear or you look over time. All right. So these are things that can definitely affect variation. All right. So please make sure you read on evolution, natural selection, genetic engineering. I really don't have the time to go through all of those, to be honest with you. Hopefully in the future I'll get to put in those in the next one. All right, so this is pretty much, we're going to conclude this review, and I thank you for being here. Thank you for just going through these points with me real quick, and I wish you good luck on the examination. I'll talk to you soon, all right?